Hello everyone, and welcome back to our biology class. In today's lecture, we are going to explore one of the most foundational topics in life sciences, the difference between prokaryotic and eukaryotic cells. We'll break down their structures, compare their sizes, and look at how plant and animal cells differ as well. Let's dive right in, the great cellular divide. All cellular life on Earth is broadly divided into two main categories: Prokaryotes and Eukaryotes. Prokaryotes. These include bacteria and archaea. They are always single-celled organisms made up of a simple cell structure. Their diameter ranges from 0.1 to 5 micrometers. Eukaryotes. These include plants, animals, fungi, and protists. They usually form complex, multicellular organisms. They are significantly larger, with diameters ranging from 10 to 100 micrometers. Core infrastructure of prokaryotes. Now, let's take a closer look at the core structure of a prokaryotic cell. 1. Nucleoid. Prokaryotes do not have a true membrane-bound nucleus. Instead, they have a nucleoid, a single, circular strand of DNA floating freely in the cytoplasm. 2. Murine cell wall, made of peptidoglycan. This provides essential structural support and physical protection. 3. Plasma membrane, encloses the cytoplasm and regulates the movement of materials in and out of the cell. 4. 70S ribosomes. These are the protein synthesis machinery, which are smaller than those found in eukaryotes, the survival toolkit. Some prokaryotes also come with optional upgrades for survival. Capsule, an extra outer layer made of polysaccharides that protects the cell against antibiotics and immune system attacks. Flagellum, a long, hair-like structure that rotates to propel the cell through its environment. Pili, short, hair-like structures used to attach to other cells or environmental surfaces. Plasmids, small, circular DNA molecules carrying advantageous extra genes such as antibiotic resistance. The eukaryotic expansion. Next, let's look at eukaryotic cells. These cells are much more complex and compartmentalized, membrane-bound nucleus. Unlike prokaryotes, eukaryotic DNA is linear and safely enclosed inside its own distinct nuclear membrane. Membrane-bound organelles. The interior is divided into highly specialized compartments like mitochondria, the Golgi apparatus, and endoplasmic reticulum. ADS ribosomes. They feature larger and more complex protein synthesis machinery compared to prokaryotes. Plant versus animal eukaryotes. Even within eukaryotes, there is a clear division between plant and animal architecture. Energy source. Plant cells use chloroplasts to manufacture their own glucose through photosynthesis. Animal cells rely on cellular respiration by extracting glucose from food they consume. Cell wall and vacuoles. Plants have a rigid cell wall and a large central vacuole for structural support. Animal cells lack a cell wall, relying on a flexible plasma membrane and internal cytoskeleton, along with small vesicles, the universal intersection. Despite these differences in scale and complexity, all cellular life shares a foundational architecture. 1. DNA, the universal genetic material. 2. Ribosomes, the essential machinery for translation and protein synthesis. 3. Plasma membrane and cytoplasm, the universal boundary network and internal cellular environment. That concludes our overview of cellular biology. Thank you for watching, and don't forget to leave any questions in the comments below.